complementary sequence to this. So when you write it out, this
Questions on the
one here is
worksheet?
gonna match onto the complementary sequence. It's this. So complementary sequence needs to have part of that as one of these to be the primer for this.
Why don't we, can you go over the compatible cookies? Let's get through the lecture here.
Okay.
And then we'll go over the compatible cookies.
So just depending on this over who's going to be the primer that will anneal this bottom. And this primer anneals that.
Let's work on this today. Let's work on today's lecture. And then we'll take some time to do the worksheet. And depending on how early we finish, maybe you guys put in your answers. So to begin today, last time we talked about, we left off midway through the process of cloning genes. So if we have our vector, in this case the vector is a double-stranded piece of DNA. So we cut, and it's got sticky overhangs probably. We want to take a piece of human chromosome, and we want to stick that into, that's my insert that I want to stick in here, and then we'll ligate that in. We'll reconnect, we'll reform the sugar phosphate backbones with DNA ligase, right? There's a couple challenges to this. First of all, if I want to take the human insulin gene out of the human chromosome and put that in there, first of all, that gene, because you guys have seen in lab, human genes are huge. There's exons that are small with very large introns. And so it turns out working with very large pieces of DNA and cloning those into a plasmid, a typical plasmid is maybe 4,000 base pairs, 7,000 base pairs if it's really large. And it's pretty easy if you've got like 2,000 base pairs to stick that in there as an insert. If you're talking 50,000 base pairs, I don't think anyone in the class is going to be able to do that. It's just very hard to work with it. If you put that in there, you put that into a bacteria, the bacteria is like, this isn't a plasmid, this is like some piece of chromosome. I don't want anything to do with this, and the bacteria won't take the plasmid up. A number of reasons why it's difficult to work with these things. So ideally, we want to work with a small piece of DNA to stick it. And the other challenge that we have, if we're going to take the human insulin gene and put that into bacteria and have bacteria express insulin, is the transcript is going to have introns and exons, all of it. And bacteria can't splice. That's a eukaryotic activity. So bacteria couldn't possibly translate that messenger RNA and get a protein off of it. So how can we get bacteria to produce human insulin? Use cDNA right here. Does it say that on there?
It just says cDNA.
cDNA.
Learning that.
So let's talk about this complementary DNA, copy DNA, some people call it. So we've got our DNA. This is the human genome here. We express gene, we make the RNA for a whole bunch of different genes on here. Gene A, B, C, D, and E. We make all of them in here, right? All these transcripts get made. And they all get processed. So I should do each one in a different color to make it easier to understand. This may take a little longer, but I think it's important for this clarity to be here. I have a whole bunch of different transcripts that then get processed into mature RNAs. So this is maybe insulin, and this is, I don't know, some other gene, some of even RNA polymerase. This is the gene that codes for something else, collagen, all the genes that you could express off of the human genome. So normally we would use this and we'd make all the proteins the cell needs to survive. But what we're going to do is we're going to break the cells open. We'll isolate all of this, and we will reverse transcribe this stuff. I'm going to show you how this works, this reverse transcription. We will learn when we get to DNA replication, and we've talked about it now already in general terms. We'll learn in great detail when we get to it, that you can only produce a nucleic acid by adding onto a 3' hydroxyl, right? You add from 5' to 3'. So what we need is some kind of a nucleotide. So what end is this, 5 or 3? This is the 5' end. What end is this? 3. This is the 3' end. So if I want to, this is the coding sequence. AUG it would be for RNA, but I just used T. So that's my protein coding sequence. What I want is to make a DNA copy that includes all of this. So do I want my nucleotide that I'm going to add on to? Do I want my first nucleotide to be here? Or do I want it to be here? Which side do I want it on? Yeah, this side. Because the 3' hydroxyl is going to be the 5' end. And this will wind up being the 3' end. And I can add onto the 3' hydroxyl and extend this way. If I had tried it this way, I did my 3' hydroxyl, I could only extend this way and I wouldn't get my code in there. Does that make sense? Okay. So what I need is a nucleotide that's going to hybridize here, and then I can extend off of. The ability, so if I take a T nucleotide and I drop that, it's going to hybridize to any A all along, any A that's in here. So how will I know that I'm going to get this? Let's say that if I take a T nucleotide, drop it in here, and it hybridizes here, and I extend off of that 3' hydroxyl, I'm only getting part of my code. Does that make sense? So what I need is a sequence. What I need is a 3' hydroxyl that I know will bind to something up here. All right? Any questions on that? So the way I could do that is I could actually look for a specific sequence in here. Let's say I get TGCA, TCCC, am I missing an N? G, T, 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 I don't want to discriminate against any. This sequence right here. Let's say this sequence is found right here. So if I made something that has a 3' hydroxyl that is complementary to that, this will hybridize right here, right? And remember this is 5 and this is 3, right? I now have a 3' hydroxyl that can go up here. What are the odds that this sequence appears somewhere else in this RNA? Low? Then we calculate it. What are the odds that sequence, if I were to randomly pick 2, 4, 6, 8, 10, 12, 15, or 13. 13. If I were to pick a random spot in here, that I would pick a spot that has 13 nucleotides going across here. That would be this exact sequence. What are the odds of that?
1 over 4 is 13.
So you had something like this in your restriction enzyme. How often does restriction enzyme cut, right? So it would be, the odds of it are 1 over 4 to the 13th, because it's 13 nucleotides long. 1 in 4 chance for each one of these, because there's 4 nucleotides. 13 long. Someone got a calculator, can calculate that out for me?
It's 1.49 times 10 to the negative 8.
Okay, so basically, for this sequence to appear, if we randomly made a 13 nucleotide sequence, and we made a whole bunch of them, we would randomly come up with this sequence once every 1.5, so let me do it this way, 1, 2, 3, 4, 5, 6, 7, 8. Does that make sense? Did I do that right? So if I randomly made this sequence up. The odds that I could get another person to come up with that same sequence randomly is 1 in 150 million. Every 150 million people that would randomly come up with a sequence would randomly come up with the same sequence I did. Does that blow your mind away? So I can actually be very specific as to where this oligonucleotide, this primer, what we're going to call it, oligo meaning many, many nucleotides assembled together, binds. Does that make sense? So it turns out, how many nucleotides in the human genome? 3.2 billion. This number is actually not far off. If I just add a couple additional nucleotides on here, I could actually randomly come up with a sequence that by odds does not appear in the human genome. And it's only about, what, 16 or 17 base pairs long. Isn't that just crazy? So easy for me to do and so unlikely to be found ever again. Okay, so I could actually get a primer that would hybridize here. I can add reverse transcriptase, and from that spot, it will work its way down and make a copy all the way down to the end. Now I have a piece of DNA, and for various reasons, I'm just going to be simple and say reverse transcriptase, when it gets to the end, says, huh, I'm at the end, that's a bummer. And it kind of nibbles this away, and then it works back and recopies what it just made, makes a complementary strand of what it just made. For the purposes of this class, that's what RNA polymerase does, or reverse transcriptase does, okay? So now I have a double-stranded piece of DNA in which my code is in the middle of it here. That's pretty cool. We can now clone one gene specifically. All I need to do is to get this sequence primer, right? Does anyone know where I can get that? Does it sound hard to get? From a catalog, right. What we're going to do is we're going to call up IDT, or I'm going to go on their website and I'm going to say, I'm going to punch this sequence in, this sequence in. I'm going to send it to them, and they're going to say, all right, it's $25, and you'll have it tomorrow, for probably 2 days. And they'll send it to me in a little tube, and then add some water because it's a powder, dissolve it, voila, got my thing. So this introduces the concept to you that if I start on this side and go this way, I can make sure I incorporate the coding sequence. I want to introduce to you the idea of a library. This is a library. All of the genes expressed are found in this group that I've isolated from these cells, right? It's a library. It's a library of all the genes humans have, right? I can convert that into cDNA, reverse transcriptase, and I can actually reverse transcriptase in a way that all of the genes get copied completely. All of the messenger RNAs get copied completely. What would my primer sequence be for all messenger RNAs? Lots of T's. If I put an oligonucleotide that is T, say 25 T's, that will hybridize to every single poly A tail. That's my 3' end, that's my 5' end. The effect would be every messenger RNA from the cell would be reverse transcribed. And now I have a cDNA library. C is supposed to be small, sorry. It's a library because all of these guys are now in DNA form. So what I have done is I have called, I don't remember, it must have been InVitrogen or CloneTech or something like that, a couple years ago, and I bought a cDNA library of from human cells. And I'll bet it cost me, at the time, it probably cost me $25,000. That was a pretty big sell to try to get. I'm pretty protective of it. Anyway, we have that. Now, out of this library, you can go and pick out your own gene specifically. And the beauty is it's already been spliced, so you need to worry about having a big honking thing cloning here. Any questions on this so far? You understand the concept of a library of genes. Good. I screwed up last year. I didn't explain it well, and people in class were like, I don't know what you're talking about, a cDNA library. I hope I've done a better job this year.
When you like buy a library of genes, is like each gene like a little powder form? Does that make sense?
They sent me a tube of liquid, and in there was a cDNA. So they had taken this, they reverse transcribed it, and then they just sold me that tube of reverse transcribed stuff.
Oh, but you still have to like get the specific.
Yeah, so everyone here in this room has been assigned a different gene. That gene exists in that library. It's somewhere in there.
Getting it out is easy.
Getting it out is easy?
Is it?
Yes. I'll show you.
Or
You're just going to get a microscope, and you can sort through all the RNAs. So any questions on this so far? Okay. So here's my oligo T primer, my oligonucleotide, a nucleotide, many nucleotides together, that's what oligonucleotide is. We call that a primer. It's a single-stranded piece of DNA, it's a primer, from which you extend off of a copy of DNA. So, this is how you will use your cDNA library. I hope everyone here is familiar with the polymerase chain reaction, PCR. This is an amazing, amazing technology. Where are we going to put this right here? The idea is, this is replication, DNA replication in a test tube. So for simplicity, we're going to have our little test tube like this. This is how I draw it. There's the cap. I think those quotes on there. In here is my double-stranded DNA of interest. For DNA to be replicated, I'm going to take this out here, the strands have to be separated from each other so that the DNA polymerase can get in and say, oh, this nucleotide needs a complementary nucleotide, and, or sorry, we'll start here. And it'll add that on, say, oh, this one, and now I use this to tell me the next one, and this to tell me the next one, and this guy to tell me the next one. They can build a copy of each strand just like that. Okay? So what we need in one of these reactions is the DNA to be able to get separated. Sorry, the DNA is anti-parallel. Right? And the way we perform that, the way we do that is we add heat. If you warm this up enough, you can break all the hydrogen bonds that are holding these guys together and they go floating apart. And then you can just take a nucleotide sequence that you want specific to your gene and add that, and that will dictate which gene in the library gets copied. We'll get to that in a second. What I want to show you is, so I have run through this reaction, I've copied the DNA one time. Now if I heat this back up, what I wind up with is this is my original strand, and this is the copy that I made off of here. This is an original strand, and this is a copy I made off of here. All of those guys can now be the template that dictates another copy. And now if I heat the reaction up again, 2, 3, 4. I can repeat the DNA replication cycle, and every one of these guys can now be a template that dictates formation of the new copied strand. sorry, it goes this way. Right? Every time I heat and then copy the DNA, I double the number of strands I have. If I start with, say, one piece of O.J. Simpson's blood in a big, huge pool of blood, you know what I'm talking about? Let's work our way through this. If I have one piece of O.J. Simpson's blood, I replicate the DNA, I separate the DNA, I melt the DNA, I heat the DNA to melt it, copy it. I now am going to wind up with 2 copies. If I melt that again and copy again, I get 4. And I double that. Next time I do it again, I get 8, 16, 32, 64. And I'm going to be lazy. I'm going to say 125. So I can have easy numbers, 250, 500, 1,000. I have melted the DNA and copied the DNA 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 times. And I now have 1,000 copies of O.J. Simpson's DNA. We can actually say 1,000, and you see the numbers are going to work out the same. We just need to add 3 more zeros down here. I now have 1,000,000 copies of O.J. Simpson's DNA. I've just run the reaction 20 times. The standard PCR, the standard reaction for polymerase chain reaction, is to run it through 30 times. To wind up with, if you start with just 1 copy, you wind up with 1 billion copies in the test tube.
Does error occur often?
Does error occur very often? Not as often as the old days, but yes, error can occur. What types of errors are you thinking of?
Just like putting in incorrect nucleotides?
Yes.
Or like shifting?
Yeah. Is the polymerase perfect when it makes copies? Absolutely not. Sometimes it puts the wrong nucleotide in. Sometimes it puts an extra one in. Sometimes it skips one by accident, so you get frame shifts and such. Yes, mistakes happen. If a mistake happens early on, that mistake will then become a large portion of the final product. If the mistake happens later, it's not a big deal. So the people who specialize in this have actually, the polymerases that do this, they've mutated them and really refined them and optimized their ability so that they make few mistakes. Millions, if not billions of dollars have been spent getting better polymerases for that purpose exactly. Other questions? Okay. So, we've just shown the power of this. We'll talk about, I think we talked about the O.J. Simpson spiel later in the semester. We need to finish this up so we get to the worksheet here today. So what we need, actually, what else we need in the reaction? So first is we need what I call a template. So that's not what I wanted. And you might want to write that down. Template is the thing we are going to copy. 1 molecule of template is enough. In theory, you want 1,000. Okay. We need some water, because everything's soluble. We need a DNA polymerase. We need a thing that's going to copy the DNA. When I have a single-stranded DNA and I start adding all these nucleotides on, what I need is those nucleotides, which are the things that actually build the DNA. So I need nucleotides, right? Do I need nucleotides or do I need deoxynucleotides to make DNA? I need deoxynucleotides. So you learned earlier this semester that R is single letter code for A or G, Y single letter code for C or T, N is single letter code for A, C, G, or T. N for anything. So I need nucleotide triphosphates, but they need to be deoxy. Right? I may regret doing that. I need something to make sure the pH and the salt environment is right. These are enzymes, so they're from living cells, so they need a certain salt environment. We call this thing a buffer. Keeps the pH in the right range. Just put it in there. And in order to copy a nucleotide, we need to have a 3' hydroxyl on which to add the next nucleotides, right? So I need a 3' hydroxyl. And that is actually going to be provided by our primer. So if I have a primer that hybridizes right here, it's going to go that way. My 3' so 3' hydroxyl is over here, so my 5' is over here. This can add nucleotides on and extend it. It'll go all the way to the end and stop. And it'll fall off. Right? So, yep, and I should have kept that in there. Out of the library of the 30,000 different cDNAs that are in that library, you want to amplify NIX. Who has NIX? NIX? Yeah. You want to amplify that. And you don't care about BCL2. So all you need to do is find a sequence in NIX, this side of the code, and say, I'm just going to use that sequence. And then I know when I PCR amplify, I will only have a 3' hydroxyl on, that attaches to my cDNA. And so you will run a PCR action, and the only thing that's going to be amplified this many times is the NIX gene. Someone else is going to design a different primer with a different sequence, specific for their gene. So out of that library, the value of having that library is everybody can use that as their template. Because everybody's gene is in there. But your PCR action is specific to you, because you, in lab, next week, are going to design the primers that you will use in your PCR action. Okay? Are there any questions on that? Okay, so I can use that to copy this strand. I need a primer to copy this strand. So do I just do this?
No.
Why not? I can't just do that? Why not? It doesn't work that way. That's not the answer I want on the exam.
Yeah, that's true.
Tell me why.
You can't add to
You can't add to the 5' end. Yeah, this is the 3' end, and this is the 5' end. This one, right? Or I guess I should label it, but anyway, yeah. So to do this, we need a primer that's going to be down here, right? If my coding sequence is in here, my protein coding sequence, I want to be on that side of my protein coding sequence, so that as I extend it down, I get all of it. Yes? So we need 2 primers. So I'll need a primer that is the same as this sequence here. Primer 1, which some people call the forward primer. And primer 2, which some people call the reverse. Some people call this sense. Some people call this anti-sense. Some people call this, oh man, left. Some people call this right. 3 different ways of saying the same thing. Let's say forward and reverse. Who wants to disagree? wants to use something different.
Can't you also call them like Watson and Crick strand? There's like
Oh my God, yes, you could probably call them Watson and Crick.
one of the.
But I think even a molecular biologist would scoff at you.
I scoffed at you too.
You're correct, though. That's probably right. All right, so we'll call it forward and reverse. Forward and reverse. So this is identical to my cDNA. This is identical to my RNA sequence, right? So T's instead of U's. Because I want that to hybridize and go this way, to the antisense strand. If I have my antisense strand, I want this one to sit like this. So it needs to be identical to the strand sequence that goes this way, right? For this strand, which is going to get copied, the primer needs to hybridize here, but it needs to be the complement of the sense strand, right? So it needs to be written, well, here's the complement of it. And you see I wrote it out this way specifically, and I chose to not erase this. Just careful about that. So that you could see that the sequence is written 5' to 3'. If I cut and paste this sequence into the ADT website, or the IDT website to order my primer, what they will send me is, they'll say, oh, he's just put his sequence in like that, and since he wrote it out, this must be the 5' sequence. Because only an idiot would send me a sequence written 3' to 5', because everybody knows you write your sequence 5' to 3'. So the actual primer I need to order is, it's written here, but it's in the wrong order, and it has to be rewritten. T, T, A, C, G, 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 A, T, G, C, A. 5, 3, right? How many of you have struggled with this on the worksheet for the primer design? You guys are liars, considering the number of people who came into my office. Struggling with that? In graduate school, I ordered primers, and I didn't do this properly. I didn't switch it over properly. So the primers I ordered were wrong, and I was never able to PCR amplify my product until I wasted probably a month of my time, and I was like, well, maybe I designed the primers wrong. Maybe it's not, a problem with the salt, or a problem with the polymerase, or a problem with the nucleotides. And I checked my sequence, and I meekly had to go to my faculty advisor, and at that time primers were about $50 each, and tell them, I need to order a new primer, I need $50 because I got to reorder. That's why I haven't been getting it this month. Embarrassing to do. So hopefully you will learn and make that mistake. If you're going to make that mistake, you'll make it in my class, or I won't be upset at you because you're learning. Okay, any questions on the PCR reaction? So the other thing we need is, we need primer 1, or the forward primer, and we need primer 2. Those are the materials that go into a polymerase chain reaction. The reaction that you will run will have 1 microliter of template. Does anyone know how big is a microliter? How many microliters in a drop of water? A small drop of water has 20 microliters. You're going to put less than 1/20th of a drop of template into your tube. And you're also going to use 1 microliter of this primer, 1 microliter of this primer. You use 1 microliter of this. You're going to use 1 microliter of this. Really small volumes. What happens if you accidentally put in 2 microliters? Any one of them. Your reaction doesn't quite go right. Probably not a disaster, but you will learn about very, very fine pipetting skills this semester. And I'll watch you guys. The first couple ones I'll watch you go, it looks like you got double the volume in here you're supposed to. You put in. Quite often people go like, oh, they'll put in like 100 microliters of my polymerase instead, which pretty much kills me because that's like $300. Lesson learned. The total reaction is going to be 50 microliters of volume. So the buffer, which comes in at 10 times strength, is going to be 5 microliters. Because when we add 5 microliters and the total volume is this, it comes down to 1 times concentration. Right? So the question is, how much water do I add? That's 5, 6, 7, 8, 9, 10. Hey, how about that? 40 microliters of water to make my total volume here. So you might want to write all this stuff down. Any questions on that?
How do you like typically design it so you like, deviate your water microwave so that it
Sometimes you might want, I won't explain why, sometimes you might want to put in primer number 3. Or sometimes the salt concentration can change whether the primer's anneal or not. So there are times that I've done double buffer concentration. And I don't know why, but for some reason DNA polymerase says, oh, okay, now it works. Didn't like it when it was a normal concentration, but you double it with salt and I'm happy. It makes my product okay. Whatever. So if I double this, I have to reduce this. Okay, so those, molecular biology is just cooking. I literally, up in my office, I have a book of recipes. Oh, you want to run this? You want to do this experiment? Here are all the chemicals that you're going to mix together. You're going to put them together and you knead them like a, no, you don't knead them like a bread, but you mix everything together and there's your reaction. Some people actually have, when I was in grad school, people would actually have a little like index card catalog. Some people used to use in the old days for recipes, like what grandma would have sitting in her kitchen. She'd, oh, I need the recipe for this, if she didn't have it memorized. They would just have all the recipes in there. It's pretty cool. Okay. So we've talked about all this. The magic of this system is, every time I heat this up, I separate the DNA strands. The problem is proteins are not stable when you heat them near boiling. Proteins denature and fall apart and are ruined. And this is a pretty cool story. So the guy who was inventing this polymerase chain reaction, I can't remember his name off the top of my head right now, who won the Nobel Prize, was at lunch. Someone, like a guest speaker, was there. So a bunch of faculty were taking the guest speaker to lunch. They went down to the cafeteria. This is a story as I've heard it. And the person was sitting there talking about, well, what I'm doing is I'm trying to replicate DNA. I'm trying to mimic DNA replication in a test tube. I'm trying to get DNA replication to happen in a test tube. And what happens is every time I heat up the reaction to separate the DNA strands, the polymerase is destroyed. So then I have to go to the freezer and take a little bit of polymerase out and add it into my reaction. Once the reaction is cool, then the DNA will actually copy, or the polymerase will then copy the DNA. And then to separate the DNA, I destroy the polymerase I added. So when it cools down, I have to go get more polymerase. And it's a complete pain in the ass to purify this polymerase. And I'll spend a month purifying polymerase so I can run 5 of these reactions. What I need is a polymerase that can handle high temperatures. And like 2 doors down from this guy was someone doing research on extremophile organisms. And that person studied DNA replication in extremophile organisms and said, wow, that's pretty cool, because in my freezer, we just purified a DNA polymerase. It is incredibly stable in temperature. I wonder if we might get this together and make this work. And it's kind of funny, they're like 2 doors down from each other and they don't know that I have the material that solves your problem, and I have the problem that makes you like $1 billion with the thing you just purified. But when someone came to their campus, they sat down together and were talking to the guest speaker about the projects. They were like, oh, hi, nice to know you. And that's how it happened. That's the story as I heard it. So that polymerase came from Thermicus aquaticus, is the name of the bacteria. We call it TAC polymerase. There's another one called Vent, and it was basically taken from one of these bacteria that live in the volcanic vents at the bottom of the ocean, handles high pressure and high temperature. There's another one out there, I can't remember, PFU or something like that. There's a couple of these very temperature stable polymerases. Yeah?
So when you heat it up to break them apart, like when it cools down, why don't they like the bonds here?
Yeah, so why don't they come back together? That's a great question. I'm glad you asked that because I forgot it, and it's very important. So we separate the DNAs. When I cool back down, these guys should just hybridize back, right? So if I have 1,000 copies of this, it turns out the concentration of the primer is such that if I have 1,000 copies of the DNA, I've got 1 billion copies of the primer. So what we have is competing reactions. So we've got DNA strand 1 and DNA strand 2 want to go back to be DNA strand 1, 2 together. But the other reaction is DNA strand 1 plus forward primer. And since I have a billion-fold excess of this reaction wins out and I get this reaction instead. Thank you for asking that question. Okay. So we need to go through these heating cycles and then cooling cycles where the DNA polymerase works. In cooling cycles, so the primer can actually bind here and extend out. So this is how we program the machine to control the temperatures. We put everything in the reaction tube, we put the reaction tube in the machine, and we say we need to melt the DNA, we need to separate the DNA strands. We do that at 94 degrees Celsius. We do that for 30 seconds probably. That's probably enough to separate the DNA, maybe a minute if you want. And then we cool the temperature down, and the temperature we cool it down to depends upon the sequence and the size of your primer. We'll talk about this in lab next time. If you have a really long primer, it's going to find its target DNA very easily at a height at 70 degrees. If the DNA is really short, it doesn't form a lot of hydrogen bonds with its target. You need a lower temperature for it to bind onto that DNA. We'll talk about this in lab next time. And then the Taq polymerase works best at 72 degrees. At the temperature we'll be annealing is about 60 degrees. It works a little bit, so it extends it out. And now, once we've grown this thing on here a bit, say it hybridized at 60, the polymerase does work at 60 a little bit, and it adds this on slowly. So now the temperature of this hybridizing is like 75. So we increase the temperature to 72, the polymerase works best and it goes real fast down like that. And this thing doesn't fall off. Did that just totally confuse everyone? So we extend the chain out, and then we go back and we melt it and do it all again. So to run the reaction 30 times is 2 to 4 hours depending on the particular reaction that you have set up. So you can run that in the morning and come back after lunch and your PCR product is done. Ready for the next step. Questions?
Is the annealing temperature just somewhere between 94C and 72C?
Yes. It's usually below 72C actually. Generally you don't want to go over 72. Polymerase doesn't work optimally. Okay, that's where I want to stop now. We'll get into Jello electrophoresis next time. I just have to keep a mental note when I write these down. It won't be on here. Let's talk about worksheets. Questions on the worksheets.
For the define the term one, where it just has like cDNA, you just want to write it
Yep.
out complementary deoxyribonucleic acid.
Yep. cDNA, complementary deoxyribonucleic acid.
Can you go over compatible cohesive ends?
Compatible cohesive ends. So I'll give you the first one. Remember I said these are not due in class today. The reason is I want to be able to go over these with you, and then hopefully you guys can do it independently or you can get together in groups. I'm fine with that. You've given it all your good shot now, and so now I'm okay with you guys collaborating and learning off of each other about this. So what was it? GATCC, right? So this is 5 prime, this is 3 prime. So the complementary sequence is going to be CCTAGG, 5 to 3, right? So first of all, let me point out to you, G, G, A, T, C, C. The restriction enzymes that we use in this lab all are palindromic like that. The sequence this way is the same as the sequence this way. Okay? Not all restriction enzymes require that, but the ones we will be using will. So I have a little notch on it like this. That means is what is it, ECOR1?
Yeah, BAMH1.
BAMH1 is going to bind to this sequence and is cut between the two Gs, which means it cuts between these two Gs also, right? So it's going to cut like this. So what we will end up with is so if I have a different piece of DNA that has a sequence of C, T, A, G, I don't care what this is, it doesn't matter what this is. We'll be done in a minute. Even though this doesn't match with this is different than this, it doesn't matter, because this G will hybridize here, this A here, this T here, the C here, and the effect is I get an A here and an A here, so what? It doesn't matter. Now all I need is DNA ligase to come in and connect the sugar phosphate backbone, connect the sugar phosphate backbone, and I have a recombinant DNA molecule. So what I'm asking you to do is for BAMH1, I diagram this out, and there's many possible answers that I gave you. You need to look at that and say, what will give me an overhang that will hybridize to this overhang? Even though this sequence here, so the sequence of this guy that cuts, what I've drawn, the sequence of this is, sorry, this is what it would cut. Oh, that's a T. Right? So if there's an enzyme, I just made this up, if there's an enzyme that cuts like this, that will work. Because there would be a T, sorry. You guys understand how that works now? The idea is I want you to think through it. It's not that you just go, oh yeah, here's the answer, here's the answer. When I sit down to grade this every semester, I'm like, oh, why did I have the students do this? I hate sitting down to figure out which of these have compatible cohesive ends. And I have to sit down and figure it all out. I actually have a little cheat sheet. The NEB catalog has a list of compatible cohesive ends and what they match up with.
So the enzyme's what's in red?
Yeah, so whatever this enzyme would be, I mean, so this is the BAMH1 binding or cutting site. And I don't know what, this is the OG1, the OG P1, the PDOG1 enzyme or something, I don't know. So that's the sequence that it cuts. And it just turns out when you cut this, and you get rid of this, this overhang matches the overhang cut on here, right? So they line up equal, you get your recombinant.
Well, what if there's no overhang? One.
What if you have a blunt end? You can ligate to any blunt cutter.
Sounds pretty
So a blunt cutter, I think the blunt cutter you guys have is TTTAAA. So what that says is TTTAAA. So it cuts like that and leaves a blunt end. So any blunt end, you can stick that back together. It doesn't matter what the sequence of the blunt end is either. It doesn't have to be A's and T's here. So blunt cutters are really useful. But sticking two blunt ends of DNA together is really hard. They're useful, but difficult to use. They're versatile, I should say. Other questions? All right, we're 2 minutes over, the last one.
For the primary pair one, you just want like the forward and like the reverse.
I want the forward and the reverse. Yep. You can slip it under my door. So I said by 5 o'clock today. I'm actually leaving at 3, and I'll be back Saturday at 1:15. So if it's under my door before 1:15 on Saturday, I'll assume it was turned in by 5 on Friday. Okay? All right. Thank you much. Have a nice weekend. As soon as the camera's off,